Good morning to everybody. This presentation is entitled A 3D Model to Study Electric Field Distribution in Homogeneous Tissue. This work is a collaboration of different research from different universities from Italy, Padova and Insubria. The electrochemotherapy is a clinical therapy used in Italy that uses electric field to permeabilize the cell membrane and enhance the effect of chemotherapy drugs. In this therapy, the electric field is applied to open hole on the cell membrane and enhance the effect the cell drug uptake by the cell. The voltage pulse are delivered to the tissue by an electrode. In non-homogeneous target tissue, the electric field is influenced by electrical property of the tissue. In this study, we analyze the effect of the tissue inhomogeneity. We perform numerical model and experiment using cell and phantom. The target of the work is the improvement of electrochemotherapy. Electrochemotherapy was introduced by Mir on 2000 and is in this work Mir described electropermeabilization to introduce non-permeant molecule in living cell, introduce gene in the cell. It's a gene therapy. It can be used in gene therapy and to ablate the tissue using irreversible electroporation. The different electroporation, reversible and irreversible, depends on the pulse setup in terms of frequency, amplitude of the voltage, and the number of pulses. The cell could be described like electrical circuit. A cell is composed by a cytoplasm, a cell membrane, and the external environment. The cell is a lipid bilayer, is a dielectric material, is permeable partially to some molecule, not all. The internal cytoplasm and external material is a conductive medium because there is ion inside this environment. The cell could be described like an electrical circuit with a resistance and a capacitance. The capacitance represents the cell membrane and the resistance the cytoplasm. A second resistance represents the external matrix around the cell. The tissue could be described like an electrical network with using this elementary model and connect a lot of different elementary models. At the terminal of this is an electric plate, this one like a capacitance. Here there is the tissue, the electrical contact. Inside there is the tissue. Is a network of resistance and capacitance. Like engineering, we can describe the tissue like only one capacitor and a resistance. We apply the voltage pulse and we have the network. Here there is the voltage pulse and the corresponding current pulse. When we apply a voltage pulse like this one, a rectangular voltage pulse, we can record the electrical current is a second, is a volt and current pulse. In electroporation, we have a set of rectangular voltage pulse that correspond a current. In electroporation, what is the effect of more pulse at high voltage? The effect is a permeability increment. When we apply more pulse, we can see an increment of the current, because increasing the number of the pole, the cell membrane permeabilization increase. The effect in conductivity is an increment of the conductivity of the tissue. This increment in conductivity corresponds to the opening of hole on the cell membrane and the possibility to include some drug inside the cell. Here we see the increment of the current with more pulse. Here there is the typical electrode used in electrochemotherapy. It's a hexagonal electrode formed by 7 needles, 6 needles on the corner of the hexagon and 1 in the center. We can apply the voltage between each pair of needle, and we have 12 pairs of needle in one electrode. For each pair, we apply a voltage pulse, and we divide this 8 voltage pulse in group of 4 pulses. The area covered by this needle is 1-2 centimeters square. Here we see it's not a large area, but we can find some problem also in this type of needle. Target tumor for electrochemotherapy, what are? We in the study of this type of therapy started at the 90 years of the century, of 20th century, and the tumor treated was at the start melanoma, basal cell carcinoma, adenocarcinoma, breast cancer recurrence on cat's wall. Oldest tumor was skin tumor or cutaneous or subcutaneous malignant tumor. With the research medical apply this type of therapy also to head and neck carcinoma and other skin tumor, skin metastasis or melanoma metastasis. At the experimental point of view, this type of therapy was used in pancreas, sarcoma, liver, and so on. What was developed, this type of therapy? Electrochemotherapy treatment was studied in Europe and started at the end of last century. In 2000, there is the first work issued by Mir, that is the researcher that developed this type of therapy. In 2006, there is issued the first guideline for electrochemotherapy. The standard operating procedure, like we see in these 2 papers, issued on European Journal of Cancer, and in this paper they describe the standard operating procedure. The electrode can be chosen, or drugs can be used. In general, they use bleomycin and cisplatin. Not all the drugs can be used in this type of technology. This 1, 2, bleomycin and cisplatin, go inside the cell using electroporation. It works very well. And the bleomycin is a very interesting. It's a very efficacy drug, but the problem was that this type of drug is not taken by the cell in normal condition. Using electroporation, the quantity of these drugs go inside the cell is increased, and cell die, because it's very effective drugs. Then this type of therapy can be improved, can be used bleomycin. And this standard operating procedure, the author described the pulse application. The pulse are applied at 5 kHz. One pulse is 100 microseconds long, and they describe the voltage amplitude applied. The general for the hexagonal needle, the 700 volt are used because the needle are 7 millimeter distant. Then the voltage amplitude depends on the needle distance. In ESOPE, protocol gives some result about cutaneous tumor. After ESOPE, a lot of tumor are studied. For instance, in 2012, different paper about different type tumor internal in breast cancer recurrence show a very, very interesting result. For instance, in case of breast cancer recurrence, the research found an overall response rate of 89%, a complete response rate of close to 60%. It's very, very interesting result. And also in other research group, they study the breast cancer recurrence result. This type of therapy could be used also in elderly patients which are not suitable for surgery or chemotherapy. What is then is a very, very interesting therapy for tumor treatment. We can see what is occur with sarcomas. Sarcomas is a very, very interesting tumor with a big variety of characteristics. That there is a lot of type of sarcoma. Some one are rich in fat, other show a myxoid stroma, other fibrous, other a combination of fat and myxoid and fibrous fat, and so on. The electrical property of tissue depends on cell size and concentration in cell type. If, for instance, they contain fat components, and on the stroma, myxoid, fibrous, and so on. Here we show you a study on sarcoma. We investigate electrical property because electrical property could influence current density, and there is another problem. For instance, there is an inhomogeneity. Inhomogeneity affect the electric field distribution, and for us is a problem. We study sarcomas. We do an experimental procedure. There is a surgery that take the sarcoma from the patient because they have to have a surgery for sarcoma therapy. And it take this mass, tumor masses, apply ex vivo the pulse, and we study, we record voltage and current, and we study also the histology of the tumor. We evaluate the resistance of, we study electrical property, and we study the histology of each tumor. We have a 30 tumor, and we see what is occurred. We evaluate, we measure voltage, current, and we evaluate the resistance. For each tumor, we have 24 voltage and current. We compute 24 resistance. We show here, we see two different things. For each tumor, there is a line. There is a difference in resistance. Each point represent one of these electrode pair. We see that different electrode pairs show different resistance inside the same tumor. For different tumor, we found a different curve with a different resistance, average resistance. Then, what is occur? What we can do? For each tumor, we compute the average resistance and we compare. We put in a graph the resistance, is the red column, for different type of tumor, and we found different class of tumor with different resistance. For instance, higher resistance corresponds to tumor based on fat. In lower resistance corresponds to tumor with myxoid stroma, and intermediate tumor resistance represent fibrous tumor. We divided tumor in different classes and we found similarity and difference. And for instance, this homogeneous tumor is fibrous based. Here there is a fat tumor based but with fibrous area. Also here is a myxoid stroma here. The other problem we found in this type of study is the inhomogeneity, heterogeneity in the treatment area. Here is a histological image at real 1x magnitude, and here there is the superposition of our electrode, the correct magnitude, on the histological image. What you see here is a different example. Here, white is fat, red is fibrous cellulate part. You see that is one needle, second needle. If you apply the electric field here, your electric field experiment to different area, fat and fibrous of cellulate. Considering not electroporated conductivity for different type of tissue, we see, for instance, that fibrous tissue is close to 0.2 Siemens per meter, is similar to collagen, and fat is close to 0.012 Siemens per meter. It's very two different conductivity. Then the electric field distribution is not like homogeneous. Here we found different type of occurrence. Here is alpha of the tissue is fat and alpha of cellulate of fibrous, all cellulate fibrous, fat, fibrous, fat, fibrous with high of fat, and so on. Then there is a lot of big variety of difference. What is occur? If here there is the simulation of homogeneous case, here the electric field, two needles, 1 and 2, there is the electric field that is distributed like this. Is not, if we have this case or this one, the distribution of electric field is not this one. This is not. We do an experiment. We take potato, is a phantom for us. Potato is very, very interesting because become dark few hours after electroporation. Not electroporated potato appear clear, and electroporated potato appear dark. We simulate the different resistivity using tissue mimic material, where we can design the resistivity of the gel, the conductivity of the gel, and we use a potato. Here is the block we used for simulation. We take a parallelepiped, divide into different regions, a gel region with a fixed resistivity, of conductivity and potato region, and two electrodes where we apply the voltage. It's a finite element model, and we found this. It's very, very interesting. If you see, it's a potato region and gel region, potato and gel with a different conductivity. And we found very, very interesting distribution. You see the distribution in this area and this area are different. And applying the same voltage, changing the resistivity, the conductivity of the gel, we found a very, very different distribution between the two cases. For instance, here, we focus on this clear blue line, and we see there is in potato region is this amplitude. If you go inside the gel in simulation, we see that is the area covered by clear blue line is smaller. The same in a different way occur in the other model. Here in potato, the area covered by clear blue line is smaller and is larger in gel region. Then there is an influence of conductivity of the material. In the same material, the adjacence of different gel with different conductivity produce a different effect. Here also there is a restriction of the area in potato of electroporation. Here we found, we applied this to a phantom model. We put potato and a gel, and we found the same result in the experimental model. In this case, that represents this simulation, the area, electroporated area, is very, very large with respect to the second one, where there is a different gel. Then, electric conductivity affects electric field distribution. The question is possible to electropore different types of tissue? What are the fast protocols to electropore? Electric field intensity is related to the cell type, the stroma, and so on. There is a correlation with the conductivity of the stroma. We need of a model that includes more variables. We mimic the cell in their environment. Electrochemotherapy experiment use cell suspension and cell adherent culture to find the parameter of electroporation. But this model are not able to simulate the complex interaction between cell and extracellular matrix. In general, in classical research, some spheroid and hydrogel are being introduced, and they are proposed to mimic the tissue and 3D dimension and the cell interaction. They are easy to handle. Not all the cells are able to form spheroid, and this model are able to mimic intercellular junction, but they are a limit in cell SCM extracellular, the link between cell and extracellular matrix. In our model, we propose a 3D scaffold for cell culture with the aim to mimic a mixed environmental found in some tube. Here I present the result we obtain in electroporation using this type of model. This type of model is a scaffold mimetic of mixed stroma. It's a cross-linked unlyophilized matrix composed of hyaluronic acid and self-assembly peptide with a laminin adhesion motif. Is a particular scaffold without collagen inside the mix. There is not collagen, but we found collagen. We prepare the scaffold, is the same image, and as an image, we seed the cell. It's easy to do. We seed the cell inside here and we culture inside few days. Here we see the breast cancer cell ECC1954, and we found what? The cell modified the scaffold. Here is the scaffold, cells are adherent, and we found this part is collagen. Cell deposited the collagen in 3 days, 5 days. Here is an example of cell culture with different cell lines from breast cancer, and we found different morphology of the tumor. Inside the same scaffold, we found different morphology. For instance, here there is a line from medullary carcinoma where cells are uniform distributed, and there is a fibrostroma, and we found cell uniform distributed on the scaffold and collagen. Here, this part is collagen. Here is different cell line is from infiltrating ductal carcinoma is this one a primary tumor, it's a metastatic tumor. Here we found a lot of extracellular matrix is collagen, is abundant fibrostroma, but also we found isle of cell. Here there is a cell with abundant collagen, cells are inside the collagen, and there is very similar to the histology of the tumor. It's an histology from a patient, and we found island or a lot of fibrostroma with not so much cell. Here there is a lot of stroma, but not so much cell. And then in the same culture we can find different area, dense in cell or dense in a cell. Here is a fast contrast microscopy of the culture after 3 days and 7 days with different composition of our scaffold. Only hyaluronic acid, EABUCAP component, and EABUCAP and ICVA with laminin adhesion motif, and we found the evolution of the culture after 3 and 7 days. We found the formation also of spheroid inside culture and the increasing of the extracellular matrix after 7 days with respect that's found at 3 days. Here there is a staining with Mason trichrome, and we found that there is collagen. Here we will see the different morphology with different composition of the 3D scaffold. Here is an experiment of PCR, and we found a collagen and laminin. Then the mass we found in the cell culture is formed by collagen. We confirm our hypothesis. Here is an image of confocal microscope where there is a staining for laminin. Green is laminin. We perform electroporation in this type of culture. We culture the matrix, the cell for 3 days. There's not so much time. We remove a part of the medium. We add the propylene iodide, that is a fluorescence dye that is able to go inside the dyed cell or electropermeabilized cell, alive but electroporated. We apply the pulse and we add each stain to mark all the cell nucleus. We use the fluorescent microscope to acquire image, and we analyze the image. Here is a sequence of voltage pulse with different amplitude, and we found very, very interesting E. Here is not electroporated cell, we are not applied voltage pulse, we increase the voltage and we see an increment of red cell, then an increment of electroporated cell. The idea of the interesting focus is here. Here is the electroporation of adherent cell with the same voltage, with the same electric field, 0, it's not so much cell, red, and 1000 volt per centimeter electric field is 10% of, not 10% of red cell, not so much. But they are electroporated in RPM, in cell culture media. This type of medium is not good for electroporation. In general, we use for electroporation in adherent cell electroporation buffer. Electroporation buffer increase the conductivity of medium and improve the electroporation. In general, with this type of field, we found a 90% of cell electroporation. But here is the electroporation of three-dimensional culture. We use also in this case RPMI. We cannot change electroporation buffer with electroporation buffer, and we found electroporation with the same electric field here and here. But here there is not so much electroporated cell, here are electroporated. What is occurring? This fact is due to inhomogeneity, because we found collagen. Collagen has a lower conductivity that is similar to the one of electroporation buffer. Electroporation buffer conductivity is between 0.1, 0.2, 0.1, 0.2 Siemens per meter. Collagen is 0.2. RPMI conductivity is 1.3 Siemens per meter. Then here, where is the collagen, we are able to electropore. Then there is the influence of extracellular conductivity. Then, inhomogeneity in tissue affects electroporation. The same applied voltage generates a difference in the distribution of electric field influencing electroporation. Inhomogeneity could affect the treatment efficacy. Thank you for attention, and special thanks to IGEA and CPA to loan the pulse generator to perform experiment. And thanks to all the authors of this work.